All right, in this video, we'll cover installing the new packages that you need for this particular lab and importing the data. So before you start, make sure that you have put those two things in your RStudio project folder that we created previously on your desktop. You'll need that week 8 code handout as well as the data file, which is this GE match 2. It's a CSV data file. So once you load that code handout, it'll pop up here for you in that R script window. Again, you can copy and paste directly or even execute the commands directly from here. But if you want, I suggest to get some practice running through it, I suggest opening a new R script, and we'll call this BIO178 Week 8 Lab. And remember to save that right away so you don't forget. All right, so the first thing we need to do this week is install some new packages, and those are the packages we're going to use to do that PCA analysis. So first, we want to install something called FactoMiner. Let me make sure I'm typing that right. That was a capital M. So we want to execute that command to install the package. While we're waiting for that, we also want to install another package called FactoExtra. This will help us accomplish the graphing, the PCA plotting that we're going to do later. Apparently this first one didn't install. There we go. Okay, and then we want to install FactoExtra. And the third package you may or may not need to install. I don't know what you've done in the past, but if you don't already have ggplot2, that's a graphing package that you will need if you don't have it already. So if you don't already have that, go ahead and install that as well. All right, so we've installed all of those packages now. What we want to do is load those libraries so R has access to them. So we will do library FactoMiner, library FactoExtra, and once we load FactoExtra, it should load ggplot as well because it's a dependency. So it's, loading, you can see down here, loading required package ggplot as well. Okay, so now what we want to do is import our data file. So first let's look at this just so you can be a little bit more familiar. So this is what our CSV data file looks like. This is set up in a way that our PCA will work. The first column here is the treatment group. So remember in that background intro video, I gave an introduction to the different field treatment groups and what the abbreviations were. That's what this first column is. So essentially every row in this data set is a different individual. And this first column here is telling us which treatment group that particular individual belongs to. And then each one of these subsequent columns here is a different gene. So these names are funky. Basically, when you do that reassembly process where you're taking those short reads and finding the overlap and reassembling that full-length sequence of the gene, the assembling programs give them weird names. So Velvet, Soap, Trinity, those are all different types of assembly programs that were used to reconstruct these full-length sequences. So that's why the names are a little bit weird, but each column corresponds to a different gene, and then the values in each cell correspond to the expression levels for that particular gene for that particular individual. So here, just as an example, that first common garden individual, for this first gene, that individual had an expression level of about 7.5 versus the next gene, it actually wasn't expressed at all, so there's a value of 0. So this is a huge data set. There's roughly 1,500 genes, so there are roughly 1,500 columns. There are, I believe, 41 rows. We have 41 individuals, and the values within the data are the actual gene expression level for each of those genes, for each of those individuals. So what we want to do is load that data into R. So let's go ahead. You can call the data whatever you want. This is an artifact of when Florian was working on it. He called it gene expression match. So that's what I'm calling it because that's what all the code says. So GE match is what we want to call this data once we load it into R. We want to read that CSV file, and that CSV file is GE match 2. So note that if you are doing this on the RStudio binder option, you're going to need to also indicate that this is in the week 8 folder. So that's indicated on the separate RStudio binder code handout that's preloaded into the cloud version of RStudio for you. So let's read in this gene expression data. So now we have that new object here, GE match, 41 observations of 1522 variables. And what we want to do next is prepare this data specifically to use for our PCA. So remember that very first column we said was just indicating which treatment group that individual was from. So we don't want to actually use that data to construct our PCA. We want the input data for the PCA to be the gene expression data itself. We just want to use that first column as an indicator to separate out which individual is from which group. So what we can do is create an active subset of our data set to use specifically to make the PCA. And if we had forgotten or just wanted to double check how many columns we actually had in our data set, you can use this n-column command or function. And if we want to see how many columns are in our data set, it'll return for us down here at the bottom, 1522. So we know we want to use the columns starting after the first one. Everything else we want to use as our active data set. So what we can do is create this new object, and we're going to specify which of that original GE match data we want to use. So we want to use all of the rows, so we don't put anything. The format is rows and then columns. If you don't indicate anything for the rows here, it's going to use all of them. And for the columns, we want to specify to use row 1 to 1521. So that's subsetting our data to gematch.active. So that's all I'm going to cover in this video, and then in the next video I will cover how to actually do the PCA and create the graphic.